Today we are going to discuss regarding the CSF, how its flow, how its work, and how hydrocephalus regarding these diseases. This is cerebrospinal fluid, and what is the composition, how amount of the synthesis, how its synthesis occur. We are going to discuss regarding these things, and we already discussed regarding anatomy of ventricles. So now I am going to start with regarding the cerebrospinal fluid. So it is like just like it's a fluid like fluid there is from the blood there is like removal of the cells and everything, and it is like a fluid. There is like less amount of like, when we imagine this is a glass, and in this glass we have put some water, and there is sodium chloride and some plasma proteins, plasma and glucose, and some number of lymphocyte, number of cell. So it is a fluid. So this is the CSF fluid. So moving on, like it's sustained, like it's suspend our it's help to the like maintain our posture, our brain in the posture, and help to like we have already also discussed regarding the function. So we discuss there. Where is made? Where is made? Where is it made? Where cerebrospinal fluid is made? So it is synthesis in like choroid plexus. It is made by the choroid plexus, choroid plexus, and where choroid plexus is situated. So first understand, choroid plexus is like a water pump or water filtering plants, like which filter it's filter and synthesis, filtered and synthesis CSF from the blood, from blood. So it is situated in the like lateral, mainly in the lateral ventricle, mainly in lateral ventricle, and minor and ventricle, and minor in third and fourth ventricle. So first I am going to rough diagram regarding this is two hemisphere, midbrain, pons, medulla, and spinal cord. So when we look, there is like two lateral ventricle, and here is a third ventricle, and after that here is a fourth ventricle, and in the posterior there is a cerebrum. So when we look in this image, there is a like, here is a foramen Monro that connecting the lateral ventricle to the third ventricle, and from this there is aqueduct. It's connecting the third ventricle to the fourth ventricle. From this there is a two opening in the lateral side, and one opening in the medial side, and which communicate with the like we already know there is arachnoid, and I am not going to draw arachnoid, not going to draw pia mater. So pia mater is attached to the central nervous system, brain, the brain. So just imagine this black is attached with the pia mater, and this is arachnoid, and we have already discussed regarding this is better to this color. This is foramen Monro, this is this is aqueduct, And here is like there is lusca, foramen of lusca, foramen of lusca, and this middle portion, this is the foramen of like Megandy, foramen of Megandy. So through this foramen, the CSF circulate in the arachnoid space. This is arachnoid matter. So there is arachnoid in the space, arachnoid space. And now we are moving on. In this, need to this color. I think better to draw in the blue color. Blue color. So in this lateral ventricle, there is choroid plexus, and also in the third ventricle. So and also in the like fourth ventricle, choroid plexus. So this is the choroid plexus, which synthesis CSF, help to synthesis or formation of the CSF. So when first we understand how it circulate. So after the synthesis, this CSF pass synthesis in lateral ventricle and passes through this foramen Monroe and go in the lateral third ventricle from both sides, and after that it passes into the into the aqueduct through in the third ventricle to the fourth ventricle, and after that it passes in the subarachnoid space by the foramen of Lusca and foramen of Megandy. It passes into the subarachnoid space. So in this space, this is known as subarachnoid space. Subarachnoid space, and in the spinal cord there is a, when we draw the spinal cord section, so there is a like posterior horn, anterior horn. In between there is a like central canal. This is a central canal. So this central canal is situated in the whole in the entire spinal cord. So through this fourth ventricle, it's communicate with the CSF. Also there is the CSF. So moving on, the when it's come in the subarachnoid space, so go into downward and go upward. It circulate in this way. This is a circulation of circulation. And when you come into the in the cranium, so moving to the drainage system. So before going to drainage, I'm going to here is like in the cranium, and in the cranium, this is a cranium, and in the cranium there is like here is the two there is two layer of the dura mater, meningeal and the meningeal. So this coming in this side and forming a here is forming supercellular dural venous sinuses. Here forming the dural venous sinuses, and when this CFS come, and here it's forming like, I'm going to draw on the image. Here this is dural venous sinuses, and when come in this region, there is like subarachnoid cells go inside this, forming finger like projection, and there is a capillary network. So this is like a pump. like cycle. It is when we do the cycle. In the cycle there is, for pumping system we put air inside, but not air pump outside from this pump with without any damage. So this type of the mechanism going on in this side. So if there is a pressure inside increases, there is a CSF pressure. CSF pressure is more than the venous. Here is the venous blood. So more than the venous, so here there is a capillary network. It's help to the pass through the CSF into this venous sinuses, and there is a drainage system. So all the coming CSF is drained out from this venous sinuses. If there is any pathology or if any of the causes increase the venous pressure more than the CSF, so there is the blockage of this pump. There is a blockage of this pump, so not through the venous to the CSF is inside the subarachnoid space. No venous blood is passing through. There is a blockage. So that the reason the CSF is drained only in this system. And if the any causes that lead to the that causes or any thrombi or inflammation or anything that causes this blockage or increase the pressure of the venous, that lead to the increased CSF pressure, and that lead to the increased intracranial pressure, or also it led to the hydrocephalus. We also discussed regarding the hydrocephalus. Now I am just regarding the how the venous drainage system is working. So this is this is the CSF, and how this is CSF is circulate in the subarachnoid space. We have already discussed. So now we are moving it's help to the brain, to float. So now regarding the function of the CSF, so discuss the function of CSF. It has 4 type of function. So first is buoyancy. It's help to the float. It help to the float the CSF, float the brain, the CSF. Brain weight is 1.5 kg, almost 1.5 kg, or 1400 to 1400 gram. This is the weight of the brain. So CSF have there is a specific gravity is tends to the specific gravity tends to the gravity. So that help to the this weight is reduced to the 15 gram. So due to the CSF, the brain CSF makes to the feel like the 15 gram. So there is no pressure on the base. When we look in the cranium, this is a cranium. Just imagine this is a cranium, and in cranium here is the brain, and in brain. So due to the CSF, the whole brain weight not lie on this lie on this surface of the cranium or the base of the cranium base. So this fluid help to the this weight is 15 gram. Due to this fluid, the weight of the 1400 gram convert into the 15 gram. So it's help to the buoyancy. There is no pressure on the base. And another function is it's help to the shock absorption. So it's like a airbag in the car, and there is a waste removal. It is like a washing machine. It's washing the whole brain in the time to time. So there is rapidly circulation occur going on. So rapidly circulation going on, and drainage, synthesis and drainage. Here is the synthesis, here is the drainage. So this rapidly 3 to 4 time in a day. 3 to 4 time in a day is a function is going on. Whole amount of the CSF is drained out. So that the reason there is what the amount of there is any waste material that is removed. So another one is the homeostasis. It keep a homeostasis. So it keep a environment or maintain pH to the maintain pH and ions balance to the whole CNS neurons. Neurons work functionally. Neurons work as finely or better function sin. So now I am going to already I have said to I am explain later on the regarding the this choroid plexus. So in the choroid plexus, like same as the this drainage system, there is like some capillary forming, like this is the forming capillary, and there is some endothelial cell, some other cell, and this, and here is the subarachnoid durameter, pia, and some here is the pumps. There is a pump. So this is like blood vessel, blood area, blood. Here is the CSF, like ventricle. So due to this, here is not permeable to anything. So there is like it's permeable to water only and some proteins. So when we look in this, there is like a pump which pumping. Here is a sodium potassium pump. Sodium pump. It is actively it using by ATP and pumping the water, and also there is a pumping the sodium. When the sodium pump, there is also chloride shifted through with the sodium, and then with this both ions, there is a water drag. So there is also passing water inside the ventricles. So there is a formation of CSF. So there is less zero slightly less than the blood protein in this CSF, and some percentage, like here is the 100 gram 100 mg of the, if we discuss regarding, here is the 100 mg of the glucose mg per deciliter. So here is like 60 mg. So two third of present in the glucose in the CSF. And regarding the composition, so when we compare with the composition, so here is the plasma. Here is like component, plasma component and the CSF component. So like sodium, when you look for the sodium, so here is the equal amount of the sodium ion, and when you look for the chloride, so there is more related more than 50%. And when we look at the potassium, so potassium is slightly less or decreased by 14%. So when we look here, there is something there is also pumping outside potassium from the CSF from the blood. There is a potassium pumping out. And when we look for the glucose, so here is less than 30% of glucose present in that CSF, and osmotic pressure is equal. Osmotic pressure is equal to the plasma. And regarding this choroid plexus, there is top of the epithelium cell, and that location is it is present in the temporal lobe of the left lateral ventricles, and also present third ventricles and roof of the fourth ventricle. So when we look, there is active transport of chloride and create osmotic gradient, and the water flows through the osmosis, and then lead to the there is a minor transport of the glucose and into the, and also outside of the sodium and bicarbonate there is potassium. this potassium and the bicarbonate and CO3 also pump out through the pump. Here is blood and here is CSF. So sodium going inside, chloride going inside CSF, glucose going glucose also going inside CSF, glucose, and also slightly proteins and some cells like lymphocyte, only lymphocyte. But from the CSF to there is a potassium coming in the CNS. There is also like bicarbonate as CO3 is coming inside the CSF to the blood. These are the how the synthesis of the CSF is occur. And after that we have already discussed the content. So now moving on the absorption, there is a arachnoid villi, and it is we already discussed, and one way flow, then pressure gradient. And regarding flow of the, we have already discussed the flow. So I am going to write the pathway for the learning process. So first there is a flow pathway. In the flow pathway, first lateral ventricle, then it passes to the third ventricle, from third ventricle through the aqueduct. So here is, I am mentioning the line, there is the aqueduct of Salvius. It's coming to fourth ventricle, then from the fourth ventricle there is a foramen flasca and two foramina flasca, foramina flasca, and one megandae, foramina megandae, and it is present in the middle, this is present in the lateral. So it's come into the cisterna magna, and then it passes into the subarachnoid space around the brain and spinal cord, and then it's go for the absorption. So it's absorbed by the arachnoid villi present in the arachnoid villi into the venous sinuses, and it is present in the sagittal sinus, venous sinuses. So this is the present in the sagittal sinus, sagittal sinuses. So for the basic information, there is cerebrospinal fluid is daily 500 ml per day is synthesis, and it is normally present of the 150 ml of CSF in this space, and it's it's rapidly the circulation, absorption and segregation. So 3 to 4 times its synthesis, and it's replaced in our day. And there is a contribution of, there is some type of cell. So in the CSF there is a contributors are there is the epididymal cell, epididymal cell which lining the ventricles, lining ventricle. We already discussed in the anatomy portion. There is arachnoid membranes, arachnoid membrane. Then there is perivascular space, perivascular spaces. And regarding this concept, we discussed in the further coming videos. So now I am moving on the hydrocephalus portion, and also there is a arachnoid belly. There is when you look arachnoid belly. So I am not going to discuss regarding arachnoid belly, perivascular spaces, and the CSF pressure how measure. So a little bit we need to understand regarding the CSF pressure, how much CSF pressure is present normally. So regarding CSF pressure, it's normally 130 mm of water, or it's compared to the 10 mmHg present when the patient is lying, when the person is lying horizontally when we measure. And there is regulation arachnoid belly and pressure sensitive valves. So when we look there is venous pressure, sinus, and the others CSF pressure, there is a slightly difference 1.5 CSF pressure with the venous pressure. Venous sinus pressure is 1.5 mmHg higher than the venous pressure. So if there is the blockage, so that leads to the abnormality hydrocephalus. So there is a blockage due to the hemorrhage, infection, congenital abnormality, and blockage, there is increase. So the up to the 500 to 600 mm of water pressure can be increased, and that leads to the increase in intracranial pressure. So when we discuss regarding these things, so there is a pathological condition called as hydrocephalus. In hydrocephalus, it is two type. One is communicating, another is non-communicating. Non-communicating. Regarding communicating, if hydrocephalus, there is a blockage in the subarachnoid space or a belly, so there is not blockage in the in this region, this foramen Monroe, or there is lateral to the not in this vector, there is not affected in the aqueduct, not affected this foramen losca and foramen of the Mengendi. So that type of the causes lead to the hydrocephalus, known as communicating hydrocephalus. And if non-communicating hydrocephalus, there is obstruction of within the ventricle system, there is aqueduct, congenital aqueduct abnormality, or there is obstruction in the cyst or tumor in this foramen Monroe, and there is effect in the foramen losca or anything that leads to the not coming the cerebrospinal fluid. So that is known as non-communicating. And in the communicating, there is an effect in these sinuses, and here is a blockage that leads to the hydrocephalus, increase the CSF fluid. So this type is known as non-communicating. So communicating hydrocephalus, obstructions, communicating, where there is an effect ventricle dilation, head swelling, and there is head brain damage, and we need to this is in the hydrocephalus lead. Now we are moving on the ending of this lecture. So non-communicating, and we have discussed regarding communicating. So there is build up extremely to and swelling. So there is nothing if the neonate in this hydrocephalus, if the patient is neonate, and neonate, there is not closure of the suture, there is not closure, so there is enlargement of the skull. And if the patient is adult or any, there is the, so in that case there is a decrease the mass of the this is the two cerebrum, and if there is a increase one lateral, so there is the, due to that this masses, it is normal. So here is the decrease in the gray matter and white matter in this left, in the right hemisphere. This type of causes are seen in the if there is a not drainage, not there is a blockage in the this like foramen of Monro. So that if there is a more CSF inside the left ventricles, so this or the gray matter, there is a degeneration occur,